Welcome to Ramchandar's Microbiology channel, Microbiology quiz on microbial diversity, Actinobacteria. Question number 1. Which of the following group of bacteria are high G plus C gram-positive bacteria? A. Clostridia, B. Cyanobacteria, C. Proteobacteria, D. Actinobacteria. Answer is D. Actinobacteria. Actinobacteria, a large and diverse group of gram-positive bacteria, are characterized by high guanine cytosine content in their DNA. These are known as high G plus C gram-positive bacteria. This means they contain significant portion of guanine and cytosine in their nucleotide sequence in their genomes. The GC content of Actinobacteria generally ranges from 50 to 74%. Question number 2. Which of the following is false with respect to Actinobacteria? A. High G plus C gram-positive bacteria. B. Rod-shaped forms to filamentous aerobic bacteria. C. Filamentous Actinobacteria produce hyphae and mycelium. D. Produce geosmins responsible for earthy smell of soil. E. They grow abundantly in acidic, marshy soils. Answer is E. They grow abundantly in acidic, marshy soils. Actinomycetes, they grow abundantly in well-drained alkaline to neutral soils than acidic, marshy soils. Actinobacteria, these are rod-shaped filamentous bacteria. They include Arthrobacter, Corynebacteria, Mycobacteria, Propionibacteria and Actinomycetes. Question number 3. Which of the following is false with respect to Actinobacteria? Some species of actinomycetes such as streptomyces, actinomyces, etc. produce conidiospores and sporangiospores. About 75% of commercially and medically important antibiotics coming from Actinobacteria. Approximately 10,000 antibiotics are isolated from species of Actinobacteria. D. Actinobacteria are cell wall-less bacteria and their gram reaction is gram negative. E. Actinomycetes found in well-drained alkaline and neutral soils. Answer is D. Actinobacteria are cell wall-less bacteria and their gram reaction is gram negative. Actinobacteria contain cell walls containing peptidoglycan and are gram positive. These are aerobic gram positive filamentous bacilli or cocci. Question number 4. Which of the following group of prokaryotes produce number of antibiotics? A. Cyanobacteria, B. Actinomycetes, C. Bacilli, D. Proteobacteria. Answer is B. Actinomycetes. Actinomycetes, a type of bacteria, are known for producing a vast majority of clinically useful antibiotics. These antibiotics are responsible responsible for producing over 2/3 of the naturally derived antibiotics used today, including streptomycin, tetracycline and erythromycin. Question number 5. In which of the following genera of Actinobacteria about 50% isolates known for antibiotic production capacity and produce over 500 antibiotics, in which more than 60 antibiotics are used as human and veterinary medicine? A. Actinomyces, B. Streptomyces, C. Micromonospora, D. Nocardia, E. Streptosporangium. Answer is B. Streptomyces. Over 700 species of streptomyces have been described. Streptomyces produce over two-thirds of the clinically useful antibiotics of natural origin, such as streptomycin, spectinomycin, gentamicin, tetracycline, chloramphenicol, etc. Question number 6. Chlortetracycline, also known as ariomycin, a broad spectrum antibiotic produced by A. Streptomyces griseus, B. Streptomyces froideae, C. Streptomyces nodosus, D. Streptomyces aureofaciens, E. Streptomyces nodosus. Answer is D. Streptomyces aureofaciens. Streptomyces aureofaciens produce chlortetracycline. Streptomyces rhinosus produce oxytetracycline. Tetracycline antibiotics, they exert the antimicrobial action by inhibiting protein synthesis in bacteria. Tetracyclines binds to 30S ribosomal subunit of 70S ribosome of bacteria, and they prevent the binding of amino acyl tRNAs to the ribosome's acceptor site and block protein synthesis in bacteria. Question number 7. Streptomycin, a broad spectrum aminoglycoside antibiotic, interfere in protein synthesis by binding to 30S subunit of 70S ribosomes in bacteria, leading to inhibition of protein synthesis and errors in protein synthesis. Is produced by A. Streptomyces griseus, B. Streptomyces froideae, C. Streptomyces nodosus, D. Streptomyces aureofaciens, E. Streptomyces naursi. Answer is A. Streptomyces griseus. Streptomyces griseus produce streptomycin, an aminoglycoside antibiotic. It binds to 30S subunit of bacterial 70S ribosomes. This binding interferes with the ribosome's ability to translate mRNA, leading to errors in protein production and ultimately cell death. By binding to the 30S subunit, streptomycin interferes with the binding of tRNA molecules to the ribosome, preventing the correct translation of mRNA into protein. This interference with tRNA binding leads to misleading of the genetic code, resulting in the production of faulty or non-functional proteins. Question number 8. Nystatin, polyene group antibiotic used to treat fungal infections in humans, especially candida infections, is produced by employing A. Streptomyces naursi, B. Streptomyces froideae, C. Streptomyces nodosus, D. Streptomyces aureofaciens, E. Streptomyces venijulae. Answer is Streptomyces naursi. A. Nystatin is derived from the actinomycete bacteria Streptomyces naursi. It was first isolated in 1950 and is a broad spectrum antifungal antibiotic. Nystatin, first antifungal antibiotic discovered by Elizabeth Hagen and Rachel Brown in 1949. Question number 9. Amphotericin. Amphotericin B, polyene group antibiotic used to treat fungal infections in humans, is produced by employing A. Streptomyces naursi, B. Streptomyces froideae, C. Streptomyces nodosus, D. Streptomyces aureofaciens, E. Streptomyces venijulae. Answer is Streptomyces nodosus. Amphotericin B, polyene group antibiotic, antifungal antibiotic. It is used to treat fungal infections in humans. It is produced by employing Streptomyces nodosus. It is a polyene macrolide group antibiotic. Question number 10. Nystatin and amphotericin B, both polyene antifungals, act by A. inhibition of cell wall synthesis by blocking chitin biosynthesis in fungi. B. Inhibition of protein synthesis by binding to 50s subunit of 80s ribosome of fungi. C. Disrupt fungal cell membrane by binding to ergosterol in fungal cell membranes and cause pore formation. D. Inhibition of fungal exocellular enzymes. Answer is C. They disrupt fungal cell membrane by binding to ergosterol in fungal cell membranes and cause pore formation. Nystatin and amphotericin B, both polyene antifungal antibiotics, disrupt fungal cell membranes by binding to ergosterol in fungal cell membranes. They cause pore formation in cell membrane, leading to leakage of cell constituents and fungal cell death. Question number 11. Which group of bacteria known for production of number of pigments such as melanin, melanoids, prodigiocin, actinobacteria, etc.? A. Cyanobacteria. B. Actinobacteria. C. Bifidobacteria. D. Bacilli, thiobacilli. Answer is Actinobacteria. Actinomycetes, particularly genus streptomycin, are well known for producing a variety of pigments. These pigments like melanin, prodigiocin, actinobacteria, etc. exhibit diverse colors and can possess valuable bioactive properties. Many pigments produced by actinomycetes also have beneficial properties such as antimicrobial, antioxidant and anti-cancer activities. These pigments are used in food, pharmaceutical industries, textiles, cosmetics, wastewater treatment and agriculture. Question number 12. Chloramphenicol, a broad spectrum antibiotic, was produced by employing A. Streptomyces frondi, B. Streptomyces nodosus, C. Streptomyces aureofaciens, D. Streptomyces venezuelae. Answer is D. Streptomyces venezuelae. Chloramphenicol is an antibiotic produced by Streptomyces venezuelae. It is a broad spectrum antibiotic used for treatment of cholera, typhoid, eye infections, bacterial conjunctivitis, otitis externa, ear infection, etc. Chloramphenicol inhibits bacterial protein synthesis by reversibly binding to the 50s subunit at peptidyl transferase and inhibit peptidyl transferase enzyme, thus preventing peptide bond formation in bacteria. Question number 13. Vancomycin is a narrow spectrum bactericidal antibiotic used primarily for treatment of serious staphylococcal infections. Antibiotic resistant staphylococcal infections is produced by A. Hemicholotopsis orientalis, B. Micromonospora purpurea, C. Nocardia brasiliensis, D. Streptomyces venezuelae. Answer is Hemicholotopsis orientalis. Hemicholotopsis orientalis, previously known as Streptomyces orientalis, is a gram-positive bacteria in the phylum Actinobacteria. It produces several substances with antimicrobial properties, including an antibiotic vancomycin. Thank you. That's all.